So this title, I'll have to explain this, I guess. It's a lovely alliteration, but there is more to it. You can buy these T-shirts. The picture is pasted together, but the T-shirts are real enough. You can buy them for £14.29 at Zazzle.com, and there are plenty of others. So one of the things I will tell you fairly soon is why it is that one of my favorite molecules is commercialized in this manner. So I'll tell you, I think what I'll do is I'll start very briefly to tell you what laminin is, and then I'll take a break and repeat what Nick said with a few pictures. So, as Nick said, my interest is in extracellular matrix, so I'll briefly tell you what this is. You all know that our cells are, our bodies consist of billions of cells that are almost entirely specialized for particular functions. There are probably hundreds of different types of cells in our body. But what is often forgotten or ignored in this view is that a good half of our body actually is acellular. And this can be fluid-filled regions in our bodies, but often this is solid material that is deposited by the cells, and you can see this here in this light micrograph between these two sheets of epithelia. You can see an amorphous structure here, which is mostly acellular. And the more extreme case is seen here on the right. This is a cell that is completely isolated from any other cell, that is completely surrounded by highly organized structures that were produced, assembled, and laid down in this very orderly fashion by this cell that sits here. And there is an unfashionable view of this extracellular matrix, which is that this is just the glue that holds our tissues together, and that it is all for mechanical stability. And while that is true, it is equally true that these cells would not know what to be if they didn't have matrix to hang on to. So these epithelial cells, if you took them away from their supporting matrix, they would very quickly die unless they became cancer cells. They wouldn't be polarized. They wouldn't know where is up and where is down. They would not be able to form this epithelium. And that is true of almost any cell in the body. There are very few cells, and the blood cells are obviously the one exception here that can live without the support of extracellular matrix. So clearly important. Now, one type of extracellular matrix that I've mostly worked on is called the basement membrane or the basal lamina. It's a bit of a misnomer. It's not a lipid bilayer, but it is a two-dimensional structure. So here's a different tissue. It's not the same tissue, but the principles are the same. So here we see a few cells originally that would have been covered by cells, but in the process of preparing this specimen, they've been stripped off. And you can see that between the cells and the matrix, so between the cells and this amorphous material here, is this carpet-like structure, and that's the basement membrane. And this supports every epithelium in the body, also the endothelia in our blood vessels, and surrounds a lot of other cell types, for example, muscle cells. So here's a diagrammatic drawing of skeletal muscle, and if you were to cut through here, strip away all the cells, and then image the remaining structure by scanning electron microscopy, you would see this beautiful extracellular material that has originally surrounded every single muscle cell. If we take a closer look at this, and this is still muscle here, and I should acknowledge this because I asked for this slide specifically because it's so pretty. This is John Heuser, who's probably the world leader in preparing these freeze-fractured images. You can see that, and again, this is an artifact of the preparation. So originally that would have been covered all the way, but it was ripped apart in the preparation. And you can see the cell surface here. You can actually even see the outer leaflet and the inner leaflet separating here. And then you can see that the cell surface is actually completely covered by this very dense network of extracellular material that is in various places connected to the cell surface. And the little nubbins here, these are all cell surface receptors that interact with these connections here. So what is this stuff here? There are a number of proteins in there. Laminin is one of them. There are others that are just as typical of basement membranes and not found in any other tissue. So these essentially define the basement membrane by their protein composition. And I'm not going to talk about these three guys here, but I will talk about laminin. Laminin is, can be extracted from certain tissues, and there's a mouse tumor that produces a lot of laminin, and this was really instrumental already in the late 70s to isolate laminin. This was long before recombinant technologies were used in this field, and so this mouse tumor yielded laminin that, when this was imaged in the electron microscope, appeared in the shape of this cross. And this is actually the reason why you can buy all these silly T-shirts. The one that I was wearing in the first slide is one of the less offensive ones. There are some really horrible ones out there. You can buy one which has a thumbprint with a laminin cross, and it says, "Every great designer leaves his mark." And so this is clearly the American Christian right going crazy on this. There are lots of them. I could show you a video by a guy called, great name too, Gilio, who gives presentations on laminin, but he has no scientific credentials. So here's laminin. One thing that these guys actually do understand is that laminin is important. Okay, so the great designer has put it into every animal. So we can go back, the fly and C. elegans, the classic model systems for genetics. But this one here is currently, I think, the simplest organism that has its full genome sequenced. So we know the full complement of protein that this sponge produces. And sure enough, it produces laminin, collagen 4, nitrogen, perlecan, etc. And so the message from this slide is, if you want to be multicellular, you better get hold of some basement membranes, because your cell differentiation is not going to work without the support of this extracellular matrix. And in flies, people, if the attachment of cells to laminin goes wrong, that has very serious consequences. This is a fly mutant, unsurprisingly called wing blister, where one of the laminin genes is mutated, and flies, I'm told, like to rub their wings together like this, and the slightest mechanical trauma leads to these blisters. And I'm showing the fly here because there is actually a human disease which is strictly analogous to this, where the slightest injury will lead to skin blistering, but the images are really gruesome, and it's a very, very horrible disease to have. And here's another one. This is a poster from the muscular dystrophy campaign. This boy has a muscle wasting disease, not due to the absence of laminin, but due to the absence of a protein that links laminin to the cytoskeleton, and I'll show which protein this is later in the talk. So clearly an interesting protein, an important protein, and although it has been studied now for over 30 years, there are still questions that remain to be answered. So this is not an emerging field, but it's a field that's been around for a long time, but hasn't had really any structural biologist work on it in a proper way, I think. So the questions that I really want to address now are, how do cells adhere to laminin? And by this I don't mean which receptors are involved, but what are the molecular recognition events that anchor laminin to the cell membrane, as we've seen. And then perhaps I'll spend more time on this, because this is more recent stuff from the lab, and I find this more exciting, is how the laminin network is formed. So these laminin molecules, as we will see, interact with each other and form a two-dimensional polymer, and we've recently begun to study this in a biochemical way. Now at this point I could skip 15 slides because Nick has done that for me, but I will not do that because there are a number of people, many of whom actually are not here tonight, but I would like to thank them because in various ways they've been important for me and in helping me come to this point. So the first ones are my parents, obviously, without whom I definitely wouldn't be standing here. Here they are. Sadly, they cannot be here tonight. This is where I spent the first 25 years of my life in Austria. A pretty picture postcard view of it, not quite as attractive in real life, I think. So this picture would have been taken at around the time when I was finishing school, and I was thinking what to do with my life. And my father was a physicist at university, and when I was little I always thought that universities seemed like fun places to work. I mean, little did I know, but I thought this would be a great place to work. So I always, in thinking about possible professions, I was probably only thinking about academic professions, so that shows my narrow-mindedness, I guess. My mother was an architect, and I like to think that some of the fascination that I have with three-dimensional structure goes also back to that, because I certainly like to do drawings a lot, architectural drawings. And the other inspiration that certainly who played a big part in my choosing this career was my uncle, Gottfried, who was a very distinguished scientist, biochemist. You can see him here. This is certainly not posed. You can see how relaxed he is. He's pointing at the mitochondria, which he had studied for all his career, and he worked on the biogenesis of mitochondria. Interestingly, he didn't train as a biochemist because when he was going to university, there was no degree in biochemistry. So he did chemistry, and he suggested I do the same. And so I started doing chemistry, even though at school this was never a subject that had interested me in the least. And I was surprised, or perhaps not surprised, to find out that I didn't like chemistry that much, actually. And I didn't like the smelliness of it. I didn't like that, it was toxic. And so it was really only in the 4th year of chemistry that I discovered a way of doing chemistry that allowed you to have a mug of coffee by your side. You could sit at a computer, and you were far away from everyone wearing a white coat. And so I really, and Birgit will attest to this, there was a time when I was really obsessed with crystallography. I just found this whole subject so interesting that I decided that's what I wanted to do. And the person who played a big part in this is this chap here, Christoph, with whom I then did a diploma thesis project. And this is my first ever crystal structure, still drawn by hand in places, of a vitamin B12 derivative. And I think many of you will know that this has great history in this country. Dorothy Hodgkin won the Nobel Prize in '64 for determining this structure. And this was my first contact with a sort of glorious history of structural biology in this country, I guess. So what is X-ray crystallography? And this is the only slide I'm going to use tonight to explain this to you. I know that the conventions of an inaugural lecture demand that I explain this to you, and I have waves and I do Fourier transforms, but I will not do this, partly because my colleague Stephen Curry did it so well 2 years ago. I think it's 2 or 3 years ago. And partly because I want to show you this is a very mature technique now, so we don't actually need to know how it works. I want to show you what you can do with it. And hence the black box, okay? So here's a crystal. You need to grow a crystal. We'll see at least one other crystal. Well, you shine X-rays on the crystal, you get a diffraction pattern like this, and you rotate the crystal, you get many of them. And then you open your black box, take out the tools of the trade and do your thing. This is not a trivial thing, but essentially it involves computers, and at the end you know where every atom in your molecule is, and that's all you need to know about it. This is the hard bit. This is actually the easy bit. Right, so as Nick said, Birgit and I moved to Switzerland. So here we are at our leaving party in Austria. We made everyone stand at this speech bubble which says goodbye, Erhard and Birgit, essentially. And we've been on this journey ever since. And it's not me that forced Birgit to go into matrix biology, but somehow our scientific paths have converged on this. She started out as a cell biologist, actually. Still is, I think. So we went to Switzerland, the Biocenter. I did my PhD with this guy here, Johann Jansonius, who was of the very first generation of protein crystallographers, so good friends with David Blow, actually, who used to work in this building. Goes way back and was a little removed from modern crystallography even when I arrived, it's fair to say. So I was very lucky that this person here, Tillman, sort of took me under his wings, and he is a very, very competent crystallographer, a nice guy as well, so we became friends. But basically everything that I know, and I think this is no exaggeration, everything I know about symmetry or molecular replacement, or all the other tricks of the trade that I haven't mentioned to you, I owe to this man here. He was a fantastic teacher, took a lot of time to sit with me and discuss textbooks, and I invited him, but I mean, he's in Switzerland, he didn't come, unfortunately. Here's my office at the Biocenter. This is a computer screen that at the time was connected to a VAX VMS mainframe computer, so no browsing here. The view out of the window was not the most inspiring either, because that was the city jail in Basel. So apart from watching the inmates exercise in the yard, which was down there, the only occupation, if you didn't want to do science, which was often, was to daydream. And that's what I dreamed about. That was my other big obsession. And actually, I have to say, one of the reasons to do a PhD in Switzerland was to be close to the mountains. That's me, and actually that's me 5 minutes before my only serious fall of my climbing career. Higher up, I was soon back where we took that picture. I wouldn't be mentioning this, perhaps I would because it was such a big part of my life, but I wouldn't be mentioning this if it hadn't had a really major influence on my career. Because one of the people I frequently went climbing with was another PhD student at the Biocenter called Patrick Maurer, and you can see him here. I think this doesn't come out very clearly, but he looks apprehensive, to say the least. He looks a little bit worried, and you might think that that's obvious why he looks worried in this situation. Not so. What he was really thinking about is how many calcium ions are there in a protein called BM40, which no one else was interested in, but that's what he was doing his PhD on, and he was getting very strange results biochemically. And so what we ended up doing is we went to our PhD supervisors, and this would have been at the end of our 3rd year of PhDs, and this was not something that I had any involvement in, and Hans Jansonius didn't like the sound of this at all because he was only interested in vitamin B6 enzymes. But we went there and said, could we please have a go at crystallizing this, because that might answer this question that was on Patrick's mind. And this turned out to be a particular stroke of luck, because this protein, I apologize, there are crystallographers in the audience, we took the Hampton screen, which was the only screen at the time, and we got hits in 25 out of the 48 conditions. So this protein couldn't help but crystallize, and less than a year later we had a structure. The answer to Patrick's question was 2. Not very exciting perhaps, but there were other reasons why this was worthy of a publication. But the lasting impact this has had on me was not this paper so much, but that it introduced me to 2 people that later became very important for my career. One was Jürgen Engel, who was Patrick's supervisor, and he's been, even though we never worked in the same lab or even collaborated after this structure, became a very, very good mentor of me, and can't be here tonight. And the other person was Rupert Timple, with whom I ended up collaborating for the next 7, 8, 10 years. And this was the man who had actually discovered laminin in '79. So he is the author of a JBC paper that's been cited many, many times more than most Cell papers, in which he described the isolation of laminin. And here he is. He's sadly no longer with us. And that's a great shot. If any one of you know Rupert, this is a fantastic shot because it really shows his character. He was the most skeptical person I've ever met. No one had a bullshit detector like him. You couldn't get anything past him. The downside of this was that he was writing pretty boring papers. So it was very much in this Germanic style of, here is what we did, we purified the protein, and it binds to ABC, and you make of it what you want. And that's how he published his stuff. He published lots of these papers, and if you ever want to be inspired, you can look up his publication record on Web of Science. His H-index is 120. So despite a truncated career, I guess. So a real giant in the field, and it was through him that I got into this field, and we ended up collaborating. And I like to think he was actually working next door to Robert Huber at the Martin Street Institute, and he chose me over Robert Huber, but that was just because Robert had 50 other projects going and couldn't be asked to do anything about laminin. So I came to Birkbeck to work on this molecule, actually. At this point, this was only a dream, really, this collaboration, so it hadn't yet happened. To work on this protein called serum amyloid P component. And this was a project with Steve Wood, who was the crystallographer, and Mark Pepys, the clinician on this project. And it was, I think it's fair to say, a very ambitious project. So the plan was to crystallize this molecule with the Alzheimer A-beta peptide, which is completely insoluble in water, or very insoluble, and wasn't ever going to form co-crystals with this. But it was a good project to get fellowship funding, and once I was there, I quickly realized that this wasn't going anywhere. So I began to really take this collaboration with Timple very seriously, and soon the parcels were arriving. He had behind him this whole factory of a Max Planck unit, so there was a styrofoam box coming in every week with purified protein for me to crystallize, which was a great way. I think back to these times, and now that we spent years making it ourselves, these were the days. So at the end of this, this turned out to be really productive, and at the end of this, of these just about 3 years at Birkbeck College, I'd accumulated enough publications and enough promise for the future that the Wellcome Trust very kindly gave me a senior fellowship. And I guess the other person then, the last person to thank, is this chap here, Henry Wellcome. If you haven't recognized him, with splendid moustache and headgear. I don't know whether he wore that to company meetings, but I think this must have been a fancy dress party. So he put the money into the Wellcome Trust, his money into the Wellcome Trust, and they've been funding me since '98, almost entirely. My entire lab is funded by them, and it's been the most fantastic time, and I really have to be very, very, very grateful to the Trust for this. Because also it has allowed me to do science in the way that I like doing science, which is with only a small group and still getting my hands dirty, and that's something that a fellowship very much allows you to do. And so finally, before we return to laminin and really stick to the science, I need to thank all the people who've worked for me in the lab. Dominic, my first postdoc, I asked him to send me a picture. He forgot, so that's what you get. Jason and Mark are now PIs in Melbourne, both of them, so they're excused. Naomi just had twins, so she's excused as well. Everyone else is here, and thanks a lot. It's been a really great time working with you. So, I'll take a sip of water, and we'll return to the questions. How we're doing. So how do cells adhere to laminin, and how is the network formed? The first thing I have to tell you to get into this is that this cross is made up of 3 polypeptides. There's an alpha chain, which goes all the way down and forms the entire globule that we can see down here. And there's a beta and a gamma chain that they are actually quite highly related, that form the other 2 short arms of the cross. So the terminology is here: short arm, short arm, short arm, long arm, which is a coiled coil of alpha helices. This is a big molecule. This is about 100 nanometers from here to here, which for a protein is large. There are 3,000 amino acids in the green chain, 2,000 in the other 2. And this molecule, and this is the sort of culmination of many, many years of devoted biochemistry, mostly by Peter Yurchenko's lab. This molecule binds via its globular domain at the end of the long arm, to the cell membrane. So this is termed anchorage. And via its short arms, and this implies that you need 3 short arms to do this, polymerizes to form this structure that, if you remember, we saw in this beautiful picture of John Heuser's. So this is what you find at the cell membrane, and the other components of basement membrane then become incorporated into this nascent laminin polymer. So this is the first step. If you don't polymerize laminin on a cell surface, you will never get anywhere with the basement membrane. So the other components are not able to do this by themselves. So I'll briefly talk about anchorage, which was the first project I worked on when I came to Imperial. In muscle, and this is different in different tissues, in muscle this is quite a complicated business, and the major receptor in muscle is a molecule called dystroglycan, which is shown in red here, which 1/2 of it spans the muscle cell membrane. On the outside it connects to the G domain, this globule on laminin. On the intracellular side it has a long linking molecule called dystrophin, by which it links to the actin cytoskeleton. And this is really an important linkage. There are other players here as well, which we don't need to mention today. This is very important. As the muscle operates, this basement membrane that is part of the muscle plasma membrane really follows all this mechanical stress. And if any part of this chain is broken, with time this leads to muscle damage, and repair is attempted by the muscle, so this doesn't immediately ruin the muscle, but with time this leads to a muscle wasting disease, muscular dystrophy. And the damage can be almost anywhere. There's a form that results from absence of laminin. There are various forms that result from aberrant post-translational modification of this molecule here. So these little wiggly lines indicate weird sugar molecules that laminin is binding to. And famously, it can result from mutations in this molecule. So this gives you the classic form of Duchenne's muscular dystrophy, which is the one that this boy we saw in the wheelchair probably had Duchenne's. That's the most common X-linked form. So we were interested in this problem here. So at the time, no one knew what these circles were, and no one knew how the recognition of dystroglycan, how that occurred. So we made this protein, and this is really just to remind me to tell you that we express our proteins in mammalian cells, beautiful red medium, and we do that not because we like it, but because we have to. These proteins are almost invariably glycosylated, so these are modifications that bacteria cannot add to a protein. They have disulfides, they often have multiple domains, and this is not something that other expression systems are very good at dealing with. So we use this human embryonic kidney cell line for production. The advantage is that it's very easy to purify your protein. It just swims in that red soup, and you pull it out by affinity chromatography, grow your crystal, and determine structure. And so this was the first structure, or actually the second, of this little bit of laminin. These two domains were revealed as a very, very common fold called the jelly roll, beta jelly roll. So you can see that these are curved beta strands. I think even as non-practicing structural biologists, you can see that these are related. These are homologous domains. There's a linker, which we don't need to talk about. And what was particularly intriguing about this structure is that there were two calcium ions. And this was intriguing because it had already been known that calcium was required for this interaction with dystroglycan. So this was great because it pointed a big fat arrow to this region saying, here be dystroglycan. And just to show the details of this, there are two aspartics, that's what calcium likes, and a few carbonyls. What calcium doesn't normally like is being sort of only half coordinated. It likes to have oxygens all the way around. And so what this implied or suggested to us is that perhaps some functional group on dystroglycan might come in and complete the coordination of that metal ion there. In the crystal, this was a buffer molecule, but that's just because it's there. So if you add dystroglycan, presumably it would latch onto this. So this was by inference, and obviously this is something you need to prove, and you prove these things by mutagenesis. So what we do is we mutate the calcium ligands and lose dystroglycan binding. I should explain this assay. Dystroglycan is put on a plastic surface. You're adding the laminin protein to it in increasing concentrations, and you detect the laminin with an antibody. It's called a solid phase assay, which we use a lot in the lab. And you can do that either with calcium, in which case you get binding, or you can put EDTA in, which strips out the calcium and indeed abolishes binding. If we mutate in one of the two domains, we lose all binding regardless of whether calcium is there or not, whereas in the other domain, this essentially looks the same as wild type. So that's telling us that this domain LG4 is the one that binds dystroglycan, and this, and I'm not going to show you this, there was a lot more mutagenesis being done, and we eventually mapped out essentially all the receptor binding sites in this business end of the laminin molecule. Now there's an obvious next step, which would be the complex with dystroglycan. It's very much the thing to do in structural biology, is to build complexes, and this is a problem because to this day, despite very strenuous attempts, no one knows what laminin is actually binding to on dystroglycan. This information here is very recent. This was a science paper last year from Kevin Campbell's group, who discovered a trisaccharide with a very funky phosphodiester here. But the question mark is where it connects to about 10 or 20 kilodaltons of unknown, presumably glycan structure. And unless this is, or until this is sorted out, we're sort of stuck with our crystallography. So I'm waiting for Kevin Campbell, who I see at meetings regularly, and I always ask him, do you know what that is? I hope that one day he works it out and we can go back to this problem. The other problem that remains unsolved is laminin's interactions with other receptors. So outside of muscle, it is actually this receptor that is the predominant laminin receptor. This is integrin, the major matrix family of matrix receptors that there is. And this binds to a different region, so this is what we just looked at. Integrins bind here, but it's probably more complicated than that. And we have a crystal structure of this, which took us some time to get and told us absolutely nothing, so I'm not showing it. This is a very tricky business because it requires probably the other chains in the laminin heterotrimer. So unless we find a way of putting all of the green stuff together, we're not going to be able to address this question. Right, so I've told you how laminin sticks to cell membranes. I'm going to now tell you how it polymerizes. And this diagram is built on a lot of indirect evidence, and a lot of it going back quite a long way. So the seminal papers here are early 90s, and the biochemistry was very beautiful, but not quite up to the standard of genetic engineering that we can do now. So as you will see, and I'm stealing my punchline here, this is actually correct, but I would argue that the evidence for this was circumstantial at best. There are some good pieces of evidence that it is these arms. So some of the muscular dystrophy mutants, there are in these arms, and the laminin is normal apart from lacking one of these globules. So that's a pretty strong indication that this is required. And there are other pieces of evidence. One is that if you throw in little recombinant short arm fragments, you can poison this process. And I'm only telling you this because that's the assay we use to probe functional interactions here. So you just polymerize laminin, which works even in the absence of cells, pellet the laminin polymer, and you just run it out on a gel, quantify it, and the amount of laminin in the pellet tells you how good it has polymerized. And then you can throw in these guys, and it completely abolishes the process. So that's how we look at polymerization. So after many years, and this was mainly Safi in my lab who I think I have to credit for this, after many years we obtained crystals of one such short arm fragments. And everyone in the laminin community had tried to do this, I think, and had failed. And it was just a long slog of figuring out which of these glycosylation sites could be mutated, which ones we could not mutate, because some are actually required for folding and secretion. So it was just a long way, and we used every laminin chain under the sun, and then this one, thankfully, crystallized. So this is the other end of the laminin molecule now. It looks a little bit like a lollipop or like a flower. So this LN domain is another one of these beta sandwiches. And the stalk, which would be a lot longer in laminin, is this ladder of disulfide bridges. So a very unusual structure if you're not used to looking at these extracellular structures. No hydrophobic core, but one disulfide after the other. And you can imagine what E. coli would do to this if you try to express this in E. coli. So here it is. But again, what does this actually tell us about polymerization? And in this case, there was no calcium ion that pointed the way, even though that process also requires calcium. So in order to progress from this, we went back to this situation here, which I explained that every animal has a laminin. And so we thought if it has a laminin, presumably it uses it for similar purposes. And by looking at sequence conservation across laminins, we might be able to pinpoint the regions that are important for this. And this worked. It doesn't always work, but in this case it came up with a very clear result. So what I'm showing you here is the surface of this molecule, two views. In orange, I'm showing you residues that are identical in worms, flies, and humans, and all other animals under the sun. In pink, I'm showing you residues that carry these sugar modifications. So there's a lot of them. Here is a conserved site that probably isn't important for function because it's right next to one of these conserved glycosylation sites. So the glycan, which is much bigger than I'm showing here, would completely shield this from any other interaction. And in fact, well, I'm not going to go into this. This isn't the site. The site that does the business is over here. It's right at the tip of the domain, so that puts it in a good place to interact with other things. It's completely free of these glycosylation mutations. And when we make mutations in this site, we can make this protein no longer bind to laminin, or to itself, rather. So this is this assay that I mentioned. If you put laminin together with calcium, it will form a polymer. About 50% will polymerize under these conditions. If you throw in wild-type short arm, so if you throw in the wild-type version of this, very little polymer forms because you're poisoning the process. And if you then throw in variants that have mutations here, they don't work as inhibitors. So you do this in a slightly roundabout way, but that's just because making heterotrimeric recombinant laminin would be quite a job. So that's why you do it in this particular way. So that is telling us that this patch, which we've identified, is binding somewhere on the laminin molecule. What it isn't telling us is where. And this is where we got a very interesting result. So we tried to use another technique that looks at interactions, surface plasmon resonance, to see what our crystallized fragment was actually binding to. It didn't bind to itself. You don't need to understand the assay here. Flat line means no result. It doesn't bind to the beta arm, doesn't bind to the gamma arm, and if you turn the whole thing around, it doesn't bind to anything either. That was disturbing. It was also disturbing because another group had published that all of these interactions occur with submicromolar affinities. We wondered about our ability to do the assay, but we could actually detect interactions. So beta and gamma in both directions do interact, but alpha seemed to be dead as a dodo here. And the solution to this came when, after a lot of head scratching, and I was sitting with Federico, as I'm sure he remembers well, at this instrument, and we were getting one line after another like this, and we were really beginning to doubt our abilities to do this. And then we just injected two things at once, and then the whole thing came to life. So I'll talk you through this because that's the key result. So this is a surface that has the beta 1 arm immobilized. Alpha doesn't bind to beta, that's the flat line here. Gamma binds to beta, but the profile here says that this is a very low affinity interaction. But when we put gamma and alpha together, you get a trace that is, even if you understand nothing about this assay, I'll tell you, is indicative of a fairly high affinity interaction. And we can do this both ways round. We can put gamma 1 on the chip and assemble the same ternary complex of alpha, beta, and gamma on the chip. The kinetics here are exactly the same. So what this is telling us that the alpha 5 arm only binds to a preformed beta gamma complex. So in diagrammatic form, beta and gamma, they do interact. We can see that in our experiments. This happens fast. It's probably transient. And what you need to form a node in this network is the keystone, which is the alpha 5 chain, which goes in there. And that really is quite a slow process. And there's a lot of funny thermodynamics going on here as well. You can disassemble this by cooling it to 4 degrees. So some really weird things must be going on here. And what I haven't shown you is that in this sensogram, if we now use one of our mutants that was inactive in the polymerization assay, this phase here where it dissociates becomes much, much faster. In other words, that complex becomes much less stable. And so the patch is probably going to sit somewhere here where it interacts with the other two chains. And this, I think, should be very obvious, is our next target. So we're trying to reconstitute this now from everything we've learned about making these fragments, and we will try and crystallize this to finally understand how the network comes together. That's a fairly ambitious target, which will keep us busy for a few years to come, I think. So in the last 10 minutes, I'll have another sip. I would like to leave laminin, much as I love it, and briefly talk about the project that Birgit and I are doing now, because that still has a lot of life in it and I think is very interesting. And this deals with collagen, which is actually the major structural protein. So in terms of dry weight, you're about 1/3 collagen. So important by abundance, at least. Now this is at higher magnification. You can see sort of structure here. It is very ordered in this axial direction. It forms these rope-like bundles and is important for tendons, for ligament, for just about every tissue in the body. It needs a collagen. We have 28, at least 28 of them. And there is, no matter how hard you look at this, there is no cross buried in this. So the designer forgot to leave his mark there. But this molecule is not endorsed by the Christian community then, but it does have a ringing endorsement from one of the founding fathers of molecular biology. Francis Crick and James Watson thought this was the most boring of macromolecules. Now, James Watson has said a few other silly things in his time, so we won't hold that against him. It also needs to be said that Francis Crick, despite this opinion, published a Nature paper in the 1950s with Alex Rich, where he determined the structure of collagen. So he clearly thought it was worth 2 pages in Nature. It wasn't just Crick, actually. There were 3 teams at the time who published at the same time published the structure of collagen. I like this best. This is Ramachandran, he of the plot. doesn't mean anything to people who are not structural biologists, but he is a name that you come across a lot when you determine protein structures. And he's looking at the alpha helix, which was, of course, Linus Pauling's discovery. And he's looking at a model of collagen here. And you can see that he doesn't seem to believe it. And he's right in being a little skeptical about this, because he actually published this a year later, so I can say this. In actual fact, the three chains are wound around each other, so they're not side by side, but the idea was right. So collagen, the basic structure of collagen is a triple helix, as I'm sure many of you will know, of three chains that wind around each other. Here's one, they're colored differently. They're all parallel, so this is not like DNA, they're all parallel. There's a stagger between the chains, but we don't need to concern us with this. And the repeating structure, this is actually not collagen itself, it's a glycine-proline-proline polymer, or decamer, that is crystallized here. The glycine needs to be a glycine because it's in the center of that triple helix. The proline can be something else, but often is proline. And in life, this second proline in each triplet would often be hydroxylated by an enzyme that requires vitamin C, as I'm sure many of you will know. In the body, these triple helices often assemble into these fibers, and I've taken this picture from a great book called The Machinery of Life by David Goodsell, which I like a lot because it gives you a real impression of how busy our tissues are. So this is the cell surface. There are some friends that we already know. So these green things are these integrin receptors. Here is actually laminin, so you can see his view of the laminin. So all of this mess is the basement membrane. Here you can see the cytosol in blue, and you can see roughly how big one of these collagen fibers is in comparison with these already large molecules that we've looked at. And each of these bars here would be a triple helix like the one I've shown you, but 1,000 amino acids long. So these are gigantic supramolecular assemblies. And cells have a way of binding to them, not in this view, because here the collagen is very far away, but there are other collagens. This would be a collagen that doesn't form a fiber that the cell needs to interact with. And there are other receptors, so apologies to Richard. There are receptors on blood cells, which I'm not showing you here. I'm showing you the two types that are most widely distributed in the human body. Again, integrins, with a special little business end here for collagen, and two receptors, two closely related receptors called the DDRs, or discoidin domain receptors, which bind collagen. And these are really very, very interesting receptors, I think. They bind collagen, as indicated here, but they're of a class of molecules called receptor tyrosine kinases that usually have no business with extracellular matrix. So this is a very strange one in the family. When these receptors bind their extracellular ligand, there's something transmitted across the membrane, which leads to phosphorylation of their kinase domains and other bits of the receptor, and this in turn leads to intracellular signaling. These molecules are important because a lot of them are growth factor receptors and are mutated in a lot of human cancers. So normally there would be a little growth factor sitting here. This would go down the route, say, divide, proliferate. With the DDRs, there are other outcomes. And another property of the DDRs that's very unusual is that they're very sluggish receptor tyrosine kinases. They take about half an hour to get aroused, but once they're going, they keep going forever. And that's totally different from all the other receptor tyrosine kinases, which click on in an instant, but then have to be quickly turned off again. And this remains a big mystery, I think, in the field. As I said, these molecules, including DDR, are interesting drug targets. This is the famous example that you always quote in grant applications. So Herceptin is an antibody against the EGF receptor HER2 and is used to treat a particular kind of breast cancer. But many others in this family are validated drug targets, and DDR perhaps is less important in cancer, although it's been found there too, but mostly in connective tissue disorders like arthritis, where your collagen gets degraded too quickly, or fibrosis, where collagen is laid down too much. And so this certainly makes this particular receptor well worthy of study. So these are the things we are interested in. How do the DDRs recognize collagen? How do they signal across the membrane? We know the extracellular portion of DDR. Again, this was a very heroic effort, which was only actually successful after we co-crystallized this thing with monoclonal antibodies that Birgit had produced in her lab. So this was, I would claim, almost as difficult as doing a membrane protein. It took us many years to get this structure. And 2 more of these beta sandwiches. I seem to be unable to get anything else. So these are jelly roll, sugars. And from Birgit's study, even before we had the structure of this, we already knew that the collagen binding site was up there. So we needed to know the corresponding site on collagen. And here's a problem. It's the sequence of collagen, of one of the collagens. You can see the repeating units here. I also need to tell you that you cannot easily express this in any way or form recombinantly. So the idea of making little bits that you express in some host and then test for binding doesn't really fly here. It's not soluble. It forms these fibers the moment you put it into neutral buffer. So at this point, the salvation, I guess is the word, came from Richard Farndale's fantastic toolkit, which is a simple idea, but someone had to do it, which is to chemically synthesize the entire collagen in sets of overlapping peptides on a chemical synthesizer. So these are 50-odd peptides, quite long, and you have to play with them a little bit to keep them triple helical. And you can then put them on a surface and use your protein of choice and essentially interrogate this, this library of peptides for binding. And DDR2 binds at 3 sites along the collagen triple helix. And Antonio, who was a PhD student in Birgit's lab, homed in on this one. As a little aside, I can tell you that the molecule that first got me into this, BM40, also binds to this region. So there's funny wheels within wheels here sometimes, I think. But he, using more peptides and alanine scanning across the entire sequence, quickly worked out that what was recognized by the DDR receptor is this motif, glycine, valine, methionine, glycine, phenylalanine, hydroxyproline, which is very hydrophobic and very different from what integrins bind to. I forgot to mention that Jonas is in the audience. He won't be offended, I hope. Jonas and Richard had a very, very influential paper in 2000 where they showed a crystal structure of integrin bound to its site on collagen, but that's very different from what is going on here. So with this information in hand, you can ask Richard to make a peptide and co-crystallize it, and this is what we did. So here's our receptor. It's only the top domain, but for the recognition, that doesn't matter. And here's collagen bound to the receptor. The receptor is shown in this surfacey representation to highlight a pocket here at the top, which takes a phenylalanine and a methionine, but from different chains. And that's neat because that explains why this receptor is not activated by denatured collagen. So you can denature collagen by heating it, then it becomes gelatin, which you all know from your cooking. But gelatin is no ligand of this receptor. So the ligand the receptor requires this to be in a triple helix. If you look, strip the surface away, the pocket is maybe now less visible, but you can see that another recognition element is actually using one of these hydroxylated prolines on the collagen. So there's a nice salt bridge that is binding to that hydroxyl here. And the reason I'm showing you this is because we weren't concerned that this structure was wrong, but it's always good to test your structures. And so you would start making mutants of this and show that it doesn't bind to collagen. And perhaps because this was all done through a chemical way rather than screening biological macromolecules, we should have really been serious about this validation of our structure. But in this case, the great designer actually came first. And in his great wisdom, he decided to create a disease called spondylometaepiphyseal dysplasia, which is characterized by very short extremities and is, I believe Birgit will correct me if I'm wrong, but I think is a proliferation defect in chondrocytes, perhaps. So your skeleton actually starts out as cartilage originally, and so something goes wrong here. And it's not just stubby arms and fingers. These patients suffer badly, and it's thankfully a very rare disease. And a lot of the mutations are boring because they just destabilize the protein, it gets degraded, never makes it to the surface. But this particular mutation, I couldn't have done better. It's a charge reversal of one of these residues involved in collagen binding. And Birgit showed that this mutant actually comes to the cell surface normally, but is completely unresponsive to collagen. So what that, I think, proves to us is that what we've done in our structural biology really replicates what's going on in the human body. And I guess at this point I better stop, but I'll tell you what we want to do with this in the future, because I really haven't told you anything about this problem. And this is a difficult one because you cannot, no one has so far succeeded in crystallizing an intact receptor tyrosine kinase. Now, lots of structures of this in isolation, of this in isolation. We have some ideas that these transmembrane helices actually interact very tightly in this receptor. That's why I'm drawing it as a dimer. But we really don't know this, and this is going to be a combination of probably very little structural biology and a lot of biochemistry and cell biology to work this out. And so finally, I take you back to this image here. This, if I didn't tell you what it was, looks like a big mess in the jungle. And I hope what I've shown you tonight is that by using crystallography, we can shine a light into some corners of this big mess. So I've told you how laminin, which may well be one of these molecules here, but we don't know, how it recognizes a particular receptor. I've shown you how the short arms of laminins like to link up in these threefold dance, I guess. And I've shown you how collagens are recognized by a particularly intriguing family of receptor tyrosine kinases. And I've mentioned some of the people along the way, so apologies to everyone in my group. There are other projects, but I think structural biology is often accused of being stamp collecting, and I've decided to only look at my 3 favorite stamps at the moment, not show you everything we've done. So there are lots of unacknowledged contributions to my research career here. So what I've decided to do is to put everyone who's ever published a paper with me on this slide. In no, well, there is alphabetic order, so this does not imply how important these people have been to me. A good third of them I've never met in my life, I should say. So the point here is twofold. Science is not done, I believe, is not done by geniuses who work in isolation. It's a team effort. These people come from all corners of the globe, which is one of the good things about science. And it does cost a lot of money. And I have already mentioned the Wellcome Trust. I better also mention BBSRC, who've given Birgit and me a grant to pursue our studies on DDR. And I also want to finish by saying that what constitutes a lot of money in this context is not a lot of money in other walks of life. So the yearly bonus of the head of one of the big banks would keep my lab going until retirement. So that puts it in perspective, I think. And I'll thank everyone here for coming tonight and for hearing me out.